So in terms of the next generation sequencing data analysis, what we are looking at is, for example, these are results, and this is a very useful link. It teaches us various tools that allows us to interpret the results. So there's something called as SAM-BAM, and SAM is the alignment is an alignment report in a sequence alignment report that is that is kind of human readable, and binary BAM is which is machine readable. So we are going to look at some aspects of this, not all, but some that are kind of crucial. So for example, if you look at the first field here, and if you look at the first line here, each of this is explained here. And for some of these, you can look at this resource, and it gives it better explanation. And once again, right now we don't have any data, but if you are working with next generation sequencing data, some of the details of these results become, it's easier to understand when you actually have numbers to work with. So I'll talk about a few of these. So the first field that is here is the same as what is called the Q name. Now remember, at, for example, FASTQ, you see that the at followed by the sequence ID tells you a lot of this information. It's necessarily going to be read, but it tells you more information about that particular read. So this Q name, this first part, is related to the information that comes after the at. Okay, so a lot of the information is built in. It's important more from the perspective of understanding what kind of information is here. The second one is what is called the flag, and the flag has to do with particular codes, and they have to do with whether the reverse sequence is aligned with the reference, whether the forward sequence is aligned with the sequence, etc. Again, this particular tool will give more information about this. The position is the starting position of the read with respect to the reference sequence. Map Q is related to the score. The log inverse of 37, negative log inverse of 37, it would give you the kind of the Fred score. Cigar we will look at in the next slide, and the 37M just means that all 37 nucleotides associated with the read match with the sequence, and that's basically it. Can get a lot more complicated, and so we can count here 1 2 3 4 5 6 7 8 9 10 11 12 13 14 15, 16, 17, 18, 19, 20, 21, 22, 23, 24, 25, 26, 27, 28, 29, 30, 31, 32, 33, 34, 35, 36, 37. So that means all 37 match to the reference. For example, and you can see the sequence here, that's the read. And we look at the qualitative and the quality scores. And remember, the quality scores are the ones we looked at here, right? The ones we looked at here. So for example, it tells you kind of a qualitative notion of exactly what that score is, what the confidence in that particular nucleotide being correctly assigned to that particular position is. And that's all we look at. It's important to begin to understand quality control. Okay, remember, this is a long process. This is a process that is getting better, but there are some built-in systematic errors because it's part of the machinery, the part of the methodology, the results associated with uncertainties, and that's why the FASTQ format, for example, gives you there's such a huge range of qualitative quality scores related to the creation, to the assembly of these reads. So let's look at what is quality control. This is kind of like FASTQ, like fast quality control, and it is essentially a report which tells you how good your experiment is. So modern high throughput sequencers can generate tens of millions of sequences in a single run, which kind of gives you a sense of the data issues that are, and that is why I think bioinformatics now is synonymous with data science, okay, and you hear the word data science, which is more and more prevalent. So before analyzing the sequence to draw biological conclusions, you should always perform some simple quality controls check to ensure that the raw data looks good and there are no problems or biases in your data. Because if there are problems, then it's one of these garbage in, garbage out deals that if you have a several problems, then you're going to get problems with your assemblies, with your reads, okay, and that then results in a lot of problems. So most sequences will generate a QC, quality control report, as part of their analysis pipeline, but this is usually only focused on identifying problems which were generated by the sequencer itself. FASTQC aims to provide a quality control report which can spot problems which originate either in the sequencer or in the starting library material. FastQC can be run in one of two modes. It can either run as a standalone interactive application for the immediate analysis of small number of FASTQ files, or it can run in a non-interactive mode where it would be suitable for integrating into a larger analysis pipeline for systematic processing of large number of files. We talked about the tuxedo suite in the last slide, and top hat, cufflinks, bow tie, etc. are part of a larger analysis, and these can be assembled into a pipeline such that the results of one become the input for the other. Here's an example of a FastQC results. And so what you see here very simply is, and here's different kinds of statistics that you can extract. So base statistics. So for example, you'll see that that once these yellow bars, and you see the error bars, which gives you the standard deviation, if they are and this is a position in the read, this is a 40 nucleotide long read, and you'll see that, for example, if these so here we have what is good quality data. So let's go to this resource and see what good quality data is. Some of the other stuff that we see in the side, we'll see what that is, and how what information is provided there. It shows that this is a universal Illumina adapter, RNA 3' adapter, etc. And we look at some of the base statistics. So it tells you what the file name is. It tells you what is good quality. And remember how I told you that if all the positions are in the green zone, whereas I suspect that this is what is a Fred quality score, which is a Fred, so, and you can see these error bars associated with it. You see everything is in the green zone, and that is obviously very good. So these are the base statistics, and we look at the per base sequence quality. That is the overall statistics. Total sequences 25,000. Sequence length is 40. The GC content, the guanine cytosine is 45. So this tells you that the score average is actually pretty good. So it's somewhere like 37. Let's look at so you can click on this per tile sequence quality. Is the quality per tile. You can look at the per sequence quality scores, and the average quality per read is 37, as I said. So you can actually look at it from a different perspective. The per base sequence count tells you, and again, if you look at our from our Fred score, A green, C blue, T red, and G seems to be the same thing. And you can see the positions, and they seem to be, there doesn't seem to be big upheavals. And it tells you that the GC content, theoretical as well as seems to be very good. It's a good bell curve. What is the N content? The N content means, remember, when we looked at the Fred score, N content is unidentifiable nucleotides are given the score of N, and you see that in all the positions there is not a single N. Sequence length distribution, okay, that is around 40. And then you have the sequence duplication levels, deduplicated sequences, and the total number of sequences. There's a small variation here, but nothing which is great. There are no overrepresented sequences. The adapter content is also 0. The solid RNA adapter content is 0, and then you have what is called the Kmer content. So in this case, what is the Kmer content? The Kmer content is kind of a region, whether it's a 2 nucleotide or a 3 or 4 or 5 region that is repeated. Okay, and so for example, in this sequence, the TACATTA, that's the repeated region, and that's what the Kmer content is. And the Kmer content, when it says as a percentage, it says something to be wary of. If there's a red X, then it's really a bad. It's really something to be concerned about. Let us now go to what we looked at, and that is bad quality data. And I think that is the one that we saw in the previous case. So, for example, if we go to bad quality data, this is what we saw, that the sequence length is 40, the GC content is 47%. But here you'll see that the Phred scores, they start at 33, and then it really deteriorates. Okay. And so you can see that the score then goes to 20 and 22, which means that 1 out of every 100. And in the good quality, you saw the per tile sequence quality was very good, but here you see all these it was just kind of a blue color. So the quality per tile is actually deteriorated. So that these are the regions where you don't see a consistency. Look at the side thing, all right? So the burr-based quality is bad. The quality is the quality scores, which is just a calculation. It's calculating it correctly. The average quality per read is all over the place. At the beginning, it's about 32%, but then it even goes, it goes to the point where you have absolutely no confidence. The per-base sequence content, again, remember you're supposed to see 4 lines that are separate, that there's no kind of an overlap. When you looked at the Phred score, you saw overlap, which means that well, there is uncertainty in some positions, for example, whatever. So for example, you see all these crossovers. So in the position amino acid 22, well, you get, it looks like there's a G content, but then it's overridden by T, which is red, and then T overrides it all. So we're never really sure what's going on here, right? The percentage G content is fine, the theoretical distribution, but it's not the bell curve doesn't, it doesn't distribute as nicely, the theoretical versus the, okay. There is between 26 and 30, there is uncertainty, okay, and that uncertainty, which means that we don't know what it is. It would be represented by an N. The sequence length distribution is fine. The duplication levels, you're going to see you see that the duplicated sequences versus the total sequences, there's a discrepancy. There's clearly overrepresented sequences, okay, which is really bad. The adapter content is not 0. There is the Illumina universal adapter, as it goes towards the end, deteriorates, and you see the Kmer content. There's all these sequence regions which see several repeats. Not a bad thing, but it's again, but it as you see in terms of the position of the read, there's some repeated Kmers here, right? And these are the counts. So you see there's obviously several of those. And that is the difference between, at a glance, if you wanted to see the difference between a good quality report versus a report which represents kind of a poor quality for whatever reason during the sequencing. There's probably some glitch in the instrumentation, etc., which gives you poor quality. Additional definitions. So for example, additional information about what some of these things mean. If you want to understand what each of these things are, you will get an explanation, right? So for example, why something is good, why something is fail. That the orange background with the exclamation mark sign is a sign of a warning, which means there's something not right. And so you'll begin to understand, if you want to see here, specifically what the problems are. Okay, so this is a good